So now we are going to see about the next topic, that is of stem cell therapy. The stem cells, we can call it as a mass of undifferentiated cells which is having the ability to capable of self-renewal and exhibit cellular potency. The meaning for this one is, in case of a plants, we are having of a cells called meristem cells. So that is, it having the ability to develop into different kinds of organ. Likewise, in human body also we having of a specific cell, it will be differentiated into numerous number of organs. That one we call it as a stem cells. The major potential of this stem cells we call it as pluripotency. So that's how we mentioned over here, that is capable of self-renewable. That is, for an example, we are taking of a lizard. Whenever a cut has been occurred in a tail of a lizard, it will be of renewable or regeneration will be carried over. But in case of human being, it's not at all possible. But a new technique is there, that is of stem cell therapy. With the help of that therapy, we can renew that cutted organ. That is nothing but our stem cell therapy, capable of self-renewable. The cutted part will be reformed and exhibit cellular potency. So what is the normal function of a cell? The same functions will be occurred with the stem cells. The second one is, it is differentiated into all type of cells that derived from the germ layer. Usually we are having of a concept called germ layer. After the fusion of a egg and sperm, it forms the zygote. The zygote will undergo several number of divisions to form a blastoral stage. From that it forms of gastrula, it forms of different layer, that one we call it as germ layer. So it is having of three germ layers, ectoderm, endoderm and mesoderm. So the major function of this ectoderm, endoderm and mesoderm is to form the different organs that present in a human body. So with the help of this stem cells, that is inner cellular mass, see here, so we are having of that blastocyst. Inside the blastocyst, a small partition has been given over, a small part has been presented over. These small parts we call it as inner cellular mass. So with the help of inner cellular mass, that's nothing but our stem cells, we can regenerate or renew the particular organ. So that is the major concept of your stem cell therapy. We are having of three different types of stem cells. One is of adult stem cells and embryonic stem cells. Along with that, we can give it of umbilical cord stem cells. But most commonly the differentiation has been given for these adult stem cells as well as of embryonic stem cells. Now we can see about it. Definitions for your adult stem as well as of your embryonic stem cells. Adult stem cells are the type of stem cells present in the differentiated tissues and the organs of adults as well as of children. That is, after the birth of a baby, these kind of adult stem cells will be present inside the nature. For an example, it may be present in a bone marrow or blood or skin. In different area or different location, there will be a presence of the stem cells. The potency, the meaning of potency is ability. The ability nature of your adult stem cells are multipotent. The meaning of multi is many. So here it will produce a different types of cells that related to each other. So again I'm mentioning about it, different types of meaning of multi is many. It will produce many type of cells, but it will be of related. For an example, I said about bone marrow. Inside the bone marrow, the process will be carried over, we call it as hematopoiesis. The meaning of hematopoiesis is the production of blood cells. So blood cells means there will be of RBC, WBCs and thrombocytes. Many cells is presented over that they are related to each other. So multipotent means different types of cells that they are related to each other. So these kinds of cells will be produced with the help of your adult stem cells. Then growing in cell culture, it is of very challenging. Why? Because the isolation of the stem cells from the human beings, then it has to be culture. For that, well equipped has to be maintained, as well as of it is of very costly or expensive. So that's why it's called as very challenging. The next one, where we can find about that adult stem cells. It may be present in skeletal muscles, livers, pancreas, brain, eye, pulp, skin, bone marrow, blood, and as well as the lining of your GI tract. So the several areas has been present for the location of your adult stem cells. The next one is of your embryonic stem cells. These stem cells are present in the early stage of your embryonic development. The previous slide, I slow one diagram. So here we are having of your ICM. So this ICM we call it as embryonic stem cells. Why? Because it is having the ability for the production of different types of germ layers. We are having of three types of germ layers. One is of ectoderm, endoderm and mesoderm. So from that ectoderm and endoderm and mesoderm, what are the organs that are present inside the body? For an example, heart, eyes, whatever it may be of, the different organs will be produced over. They are not related to each other. That kind of potency we call it as pluripotent. Okay, so the function of your embryonic stem cells is of pluripotent nature. Here in case of adult stem cells, it having a multipotent. It will produce the cells but they are related to each other. Here it also produces different kinds of organs or cells but they are not related to each other. Each and every is having a specific function. So the culturing of these stem cells is also easy. Why? Because it has been isolated from the embryonic cells. So the embryonic cells is having an ability to divide, that is proliferation and differentiation will be easy. The meaning of proliferation and differentiation is single cell will be divided into two cells, two will be converted as four. Likewise the divisions will be carried over. Later on it will be forms an organ. I think the level of organization is easy to understand about this culture. One is the cells aggregates to form of tissues, the tissues aggregate to form of organs, organs aggregates to forms of system. Likewise here also aggregations will be very easy. That's why it has been considered a relatively easy one. The examples where it has been present of ES cells, it's present in the inner cellular mass of a blastocyst. So this one we call it as ICM, inner cellular mass of a blastocyst, which give rise to the three germ layers. So here it has been depicted about your potency of your stem cells. In that previous slide I have mentioned only about two things, that is of multipotent and pluripotent. Here we are having of additional to that totipotent, oligopotent as well as of unipotent. So the first two comes under the category of your embryonic stem cells and the remaining three comes under the categories of multipotent. Totipotence, ability of a single cells that developed into a whole organs or a whole body, we call it as totipotency. Pluripotence means is differentiated into cells from any of the three germ layers, that is of pluripotent. Multipotent, it differentiate into a limited range of cell types, that is of multipotent. Oligopotent, it represent differentiated into a limited number of cell types. Unipotent, it differentiated into a single type of cells. Uni means single, so it was differentiated into a single cell. The meaning of differentiation is maturation. So after division, what happened? A single cell will attain a maturation. That maturation we call it as differentiation. So these are the potency that has been followed in case of your stem cells. So with the help of these stem cells, the therapies has been carried over to cure the diseases. So how we can store of your stem cells? The stem cells can be stored with the help of banks, that is stem cell banks. Extraction, processing of and storage of stem cells, we call it as stem cells banks. So you're having of several steps that has to be followed in case of your stem cell banks. As like of your egg storage as well as of sperm storage, here also the stem cells is ability to store into your bank with the help of a process called cryopreservation. So cryopreservation is a step, is an important procedure that we have to follow for storing of a stem cells. First, it has to be collected from the umbilical cord of a baby. After the parturition, that is birth of a baby, within 5 to 10 minutes, umbilical cord has to be cut. The meaning of umbilical cord is a connection, a tube between a mother as well as a fetus, we call it as umbilical cord. In that, the inner cellular mass, that is embryonic stem cells, will be presented over. After the birth of a baby, that is within 5 to 10 minutes, it has to be cut safely with the help of a doctor. So it has to be cut. From that, the stem cells has to be used, it has to be collected and it has to be cryopreserved before it has to be checked to evaluate the sample quality. The meaning of cryopreservation. Preservation, we all know that it is of storage. Cryo means at low temperature we are going to store the particular stem cells. low temperature, it depicts of minus 196 degrees centigrades. So cryopreservation is freezing and drying. Simply we can call it as freezing and drying. So with the help of low temperature, that is of minus 196 degrees centigrades, we can store the stem cells, and later we can use it for the therapy, that is culturing of the cells. A single cell will be cultured. So the single cell is having of different potency. So much potency will be presented over. So with the help of a potency, it will be differentiated into any one organ. So an organ can be renewed with the help of a stem cell. So this one we call it as a stem cell therapy. The steps that has been important over. The next topic we call it as molecular diagnosis. The meaning of diagnosis is finding about the presence of infection that present in a human body. But in a traditional method, we can diagnose the infection only after the occurrence of symptoms. When a man or a human getting of infection, what happens after a few days or few hours, the symptoms will be slowly occurs. Then the samples will be collected from the patients. Then it will be of diagnosis which kind of infection it was. Either it may be a bacteria or a viral or a fungal. But yearly diagnosis is important for making a cure of our diseases. That can be done with the help of a method called molecular diagnosis. Yearly detection of disease can be done with the help of an rDNA technology. So in this chapter we are having only of two things. One is of PCR as well as of ELISA. PCR, polymerase chain reaction. ELISA, enzyme linked immunosorbent assay. So we are going to make a discussion based on ELISA as well as of PCR. First thing, that is concentrating on ELISA. As I said before, the abbreviation for your ELISA is enzyme linked immunosorbent assay. So the meaning of an enzyme, it acts as a catalyst. So one reaction has to be occur means there must be a mediator. Simple word, we call it as a mediator. It will involve in the connection of two components. It's linked the immunosorbent assay. So immunology. Immunology means we have to remember about two things. One is of antigen and antibody interaction. Antigen antibody interaction along with the enzyme and the substrate interactions, we are going to make one diagnostic method. We call it as ELISA. So here a combination of enzymes and substrate so that it forms a coloration, antigen and antibody so that it forms a reaction. Two method has been combined together for making of yearly detection of an infection. This particularly this ELISA is used for detecting of a disease called AIDS. It was discovered by the scientists Eva and Peter. So here we having of different types of ELISA. One is of direct ELISA, indirect ELISA, sandwich ELISA and competitive ELISA. Mostly all the ELISA's technique is having of a common procedure. Slight deviation has been presented over to differentiate it into a different name. Now we are going to discuss about only one thing, that is of sandwich ELISA. The meaning of sandwich is a combination of many. So here also you are going to use of different kinds of components to find out the early detection. The first one, we are going to make one well. We call it as micro titer well. Nothing but a slide. It is having of different dots. So in that we are going to add of a sample. So patient has come. We are going to collect the sample from the patients. That sample has been placed in a box which is having a small wells. So here this one is considered as a well. The pink color is called as a sample. Then the second step, it has to be added of an antibodies or an antigen. Either it may be replaced by antigen or antibody. First the antibody may be attached, otherwise an antigen may be attached. The thing is either one has to be attached over. The antigen will comes and bind of your antibody if it is of specific. As I said before, antigen antibody interactions will be carried out over here. The next step is the secondary antibodies will be added over. So this yellow color thing we call it as an secondary antibody, which will be go on attached with of your primary antibody. Along with that, it is having of one red color dot, we call it as an enzymes. Whenever we are adding of a substrate, these enzymes will go on react with of a substrate so that it forms a complex. So the product will be of colored. So what happened? A blue as well as of a pink color has been mingled so that finally it forms a colored product that is of yellowish. So this is an example. So with the help of this color formation, we can identify that the sample is having of that particular infection. If there is no color formation at the end of the procedure, we can simply say that the particular patient is not infected by any kind of diseases. So this is the simplest way to detect the infection at the early stages. This is about your ELISA. The next one is of polymerase chain reaction. So polymerase chain reaction, the simple meaning for your polymerase chain reaction is conversion of a single copies of DNA into multiple copies of DNA. Conversion of single copies of DNA into multiple copies of DNA. The name itself is having the meaning polymerase chain reaction. The meaning of polymeration is production of more amount of DNA chain. Continuously the productions will be carried over. So it has been called as PCR, polymerase chain reaction. A technique in molecular genetics that permits the analysis of any short sequence of a DNA, even in the sample containing only a minute quantity of DNA or an RNA. So along with of ELISA, the PCR is also used in case of early diagnosis of your infection. Even though we are having of a small amount of sample also, we can use of this method to diagnose the particular infection. So here we are going to follow about 3 steps. One is of denaturation. The first step we call it as denaturation. The second one is of annealing, and the third one is of extension. Remember that we are going to use of 3 things. One is of denaturation, another one is of annealing, and the third one we call it as extension. So the steps has been given in this diagram. So this blue color we call it as a double stranded DNA. Usually the DNA will be in double stranded for their stability. So in between of the double stranded we are having of a bond. The name of the bonds we call it as hydrogen bond. So it forms of a double structure. The first step in case of your PCR is denaturation step. The meaning of denaturation is we are going to convert the double stranded DNA into a single stranded DNA. That is the name, we call it as denaturation. How is possible? By giving of an heat. So we are going to provide an heat about 94 to 95 degrees centigrade. Automatically the bond that present between of that 2 DNA will get break down. Automatically it forms of 2 single stranded molecules. Immediately it goes for the second step, that is attachment of a small sequence of DNA, otherwise an RNA primers will be added to it. The meaning of primer is a small DNA sequences is added to it, or RNA sequences has to be added to it. So this is the step that has been followed in case of annealing, that is attachment of primers to the separated DNA molecules. What is the uses of this attachment? What happen? It will be act as a inducers. So as I said before, with the help of a single copy of DNA, we are going to produce of multiple copies of DNA. See here we are having of 1 DNA. At last, at the step we are getting of 2 DNAs. How it's possible? First, it has been separated as 2 strands. Second, a primer has to be added to it. Along with the primer, enzyme has to be added along with it. That is of a specific enzymes. We call it as tag polymerase enzyme. The name TAQ, tag polymerase enzyme. Polymerase, that means amplification, that is attachment of your DNA, the formation of a new DNA, we call it as polymerization. Tag is the name of the organism, is a bacteria, Thermus aquaticus. It is a bacteria from which the particular enzymes has been taken for or isolated for. So the specific thing is it will be of heat resistance. It can be surveyed up to 95 to 100 degrees centigrade. So the particular enzymes will be used over. So with the help of that enzymes, the DNTPs, that is here we are having of a rule, that one we call it as Chargaff rule. One strand is having of adenine, means the nearby strand should having of thymine. If it is having of guanine, means it should be having of cytosine. Likewise, with the help of the tag polymerase, the addition of your DNTPs will be carried over, so that at last the extensions will be done. It forms of a 2 double-stranded DNA. That is the last step. It will be done with the help of a temperature called 72 degrees centigrades. So these are the steps that has been followed over in case of a PCR. Again, I am repeating that. So in PCR, we are having up 3 steps. One is of denaturation, second one is of annealing, and the third one is of extension. Denaturation is conversion of a double-stranded DNA into a single-stranded DNA. Then annealing is for attachment of a primer. Extension is for the polymerization, that is synthesis of a new DNA strand. So with the help of this, we can diagnose the disease-causing pathogens that present in the human samples. The advantages for your PCR is it will be ability to amplify a sample of DNA When are there, it's not enough. That is, limited amount of sample is enough for making of an diagnostic things. It is used for the samples that occurred from a crime, or archaeological samples may be also used as. It is an important tool that has been used for medical diagnosis. It is used in genetic testing, that is to find out the mutation, presence of mutation that present in a sample. Then it is also used in genome sequencing. Genome sequencing is a step that has been undertaken in the process called Human Genome Project. The major function of Human Genome Project is to find out the location of a gene that present in a genome. Genome means a group of genes, we call it as genome. The function of gene is to produce a protein. So with the help of this Human Genome Project, we can find out the location of a gene that present in a genome. So after finding of their location, easily we can identify the function of that particular gene. So for these and all, the PCR will be of advisable to making of its advantages.